Dobré ráno, dámy a pánové. Good morning, ladies and gentlemen. I will have some unusual role today to introduce our speaker. Our speaker is Martin Kolisko from University of South Bohemia in České Budějovice, and he will talk about a new software for phylogenomic analysis.
Okay. Good morning. Thank you for the introduction. My name is Martin Kolisko, and I'm going to be talking about a software tool that we just recently published called PhyloFisher. It's basically a software for semi-automatic phylogenomic dataset constructions. You'll see why semi and not fully automatic in a little bit. And I just want to say that this software has been in development for years. It started during my PhD when we just kind of write a very dirty and quick script, and it slowly kept growing and growing into this bigger software until my colleague Matthew Brown from Mississippi State University decided to make it an actually official software and publish it. So I'm just going to start a little bit wide this introduction of phylogenomics, and the reason I'm doing it is that I'm not sure that all of the audience understands exactly what it means, and also to show what are the hardest parts of phylogenomics and how our software is basically helping to solve them. So phylogenomics is basically a phylogenetic analysis of sequence data where you use multiple genes to infer the organismal relationships, usually tens, hundreds, even thousands today. And it's slowly become kind of the bread and butter tool for many evolutionary analyses, whether we just positioning a single lineage on the tree, which in past would be done by a single gene, or it's also really important to compute trees for comparative analyses because we need a good evolutionary framework for those. And the problem is that when we use single gene phylogenies, which are much easier to perform in phylogenomics, there is an ultimate problem that generally any single gene doesn't contain strong enough signal to fully resolve the phylogeny. The problem is that within the alignment, there's a lot of noise and only a little bit of the true phylogenetic signal. The idea of phylogenomic is that if we take multiple genes and put them all together, the true signal of each of the genes get combined together into a stronger signal, while the noise just remains a noise, and we get a better resolved tree. And overall, it works very well. There is obviously caveats, and we're going to talk about them in a little bit. But as I said, it's a very powerful method, and it's used a lot. And this is just an example of a few recently published papers in very high impact journals. And the main problem with phylogenomic is that it used to be the tool of the few experts that really know how to do it, and that really know how to assemble a data set and how to analyze the final version of a data set. Right now, it's basically a tool for everyone. Many people use it that have only partial knowledge or skills to do it correctly. And that's what we're trying to solve with this software. If you look at the procedure for phylogenomics, it's very simple. We choose the organisms, get their data set, whether it's genomes or transcriptomes. We identify the data we want to use. We get rid of paralogs because we only want to use orthologs, as I explained a little bit. And we do alignments, trimming maybe of the alignments, depends on the software, and then we compute a really nice phylogeny. Afterwards, we do some estimation of statistical significance and potentially other analysis to test for different things. And the really most crucial part of the dataset assembly is identifying paralogs and other problematic data, and I'll explain why. So I don't know if how familiar are you with Paralogy. I assume you are at least a little bit. Basically, Paralogy happens every time a gene duplicates, okay, like it happened here. And from the point of view of a phylogenomic dataset assembly, we can think of 3 different versions or kinds of paralogy. One is like this. It's the deep paralogs. Okay. For us, as when we do phylogenomics, we can treat these in 2 different genes, basically, because it's an ancient duplication that happened before the radiation of the organisms that we're trying to analyze here. They are fairly simple to identify. It's not such a big problem. Then there are what we call in paralogs. Those are basically gene duplication only within one organism on the tree. Again, here, whichever of the sequences we choose, it doesn't matter. So it doesn't really pose a problem for selection. But there are these so-called, what we call them, mid paralogs, which is basically a situation when the gene duplication happened only for some part of the, so one part of the organisms on the tree. Like here, it happened for animals, and here we really only want to include sequence data from either the paralog 1 or the paralog 2. We never want to combine the 2 together. And well, it's because if we do that, if we would do it, for example, like this, the final topology for this gene would look like this. It would be the wrong one. And while for other genes we would, let's say, didn't have this problem, or we would do the selection correctly, we create a lot of conflicting signal. Okay, and that's a problem. And you know, this is actually a relatively new problem. In the past, we knew that selection of orthologs is really important, but they were kind of saying, okay, well, we have hundreds of genes. If we mess up a few of them, it doesn't matter because the signal from all the other ones will override the wrong of this particular gene with the red arrow, and we get the right answer. Well, it was true until people start actually looking into it and find out that that's not true. That, you know, a little bit of paralogy, a little bit of a wrong signal in the phylogenomics can totally change the topology. And not just change the topology, but change the support. Here in this paper, they tested this and find out that as much as, you know, 1 or 2 genes added or removed with, for some reason, conflicting data, the support for a certain group could drop down from, you know, 80, 85 for one type of relationships to a support of 80, 85 to a different kind of relationship. So very few genes make a very big difference, which made it very clear to us. And the people that work in this, they were realizing this more and more often, because more and more we discovered that we have mistakes in our trees, that we have paralogists included. We find out it actually makes a difference in the final trees. So, you know, more than ever, the truth is garbage in, garbage out. So we have to love our data. We have to, you know, take really good care on selecting orthologs and paralogs. And I'll basically, with PhyloFisher, I think we solve this problem in a very crude way, but very efficient way, as you will see lower. The other part of phylogenomics that tends to be very often problematic for people who are not well versed in this kind of analysis is, what do you do after you're done with the dataset assembly and when you compute the first phylogenetic tree? And that you have to be careful and you have to check for possible errors, for systematic errors. If I go, unfortunately, I didn't make this. If I go back up really quickly, accept my apologies for it. Yeah, if I go back to this this image, I said that here with phylogenomics, we amplify the true signal to override the noise and get a good topology. That works very well until we have some sort of systematic bias in the genes. And when we have the systematic bias in the genes that we don't have a good way of getting rid of, we will amplify the systematic bias by adding more and more data. It's a systematic error. So more data we throw at it, the more support we get for the wrong answer. And you know, there's quite a few common mechanisms like long branch attraction artifact, which basically means that fast evolving lineages will tend to branch together on a tree if our model is not good enough, and which is almost never true. So very long branches on the tree always pose a problem. And with phylogenomics, because we have a lot of data and because there's a systematic error, even a little bit of the wrong data might cause a problem. So basically, every time you do phylogenomic analysis, people that do it they should do these checks. They should look for gene resampling. They should subsample their genes. If they have 100, 200, they should divide 20, 40, 60, 80, and randomly resample them into a group of 100 genes, and then recompute them and see what they get back. Do the support for the key relationships on their tree change or not, right? And here we can see that some lineages defined here on the right receive very high support with 20 genes, with 40 genes, 80 genes all the time, while some of them, the more problematic relationships, they don't. They do get a very high support here, and the conflicting topology is very low support here with a lot of genes. But and we have less genes, we can see that it varies. But everyone should do this to make sure that there is not a strong selection bias due to selection of genes or anything. Here we think it's fine because here this is just purifying the signal out into one thing. Then here, the other tool people usually use is fast evolving site removal. That's a tool that should help see whether we do actually have an effect of long branch attraction artifact, because we are slowly removing the worst kind of data that would cause the long branch attraction artifact, the fast evolving sites, and we remove them by little badges. Here we remove them by 1,000, and you can see that one of the relationships, the one we are actually studying in this paper, it looks very good. It's very high here in the full data set, but as we start removing the badly behaved data, the fast evolving data, fast evolving sites, it goes down. So we know that this topology, this particular relationship is not very reliable, and that should be done every time just to confirm that what you present in the results is actually supported, and it's not only supported statistically, because here we have a very high support, but it's also not changing as we changing the data set, as we're removing potentially bad data. The other problem with phylogenomics today is the sharing of data and publication of data, because very often people don't share the raw data. They would share their, let's say the sequence files or the assembly of each of the genomes or transcriptomes, but they don't share the actual phylogenomic matrix at all. Sometimes they just share the whole concatenation of the hundreds of genes. Sometimes they just share trimmed single gene trees, single gene alignments, which are useless because they're unusable for any downstream analysis, and we cannot check whether their decision, for example, in the ortholog parallel selection was correct. At best, what people share is untrimmed selection of orthologs, and this is a problem, and I'll show you why. So imagine we were assembling a phylogenomic data set, and here is our, on the left, is our initial tree with all the sequences we are looking at. The set of sequences we're selecting orthologs from, the final sequence for data set. And the red stars are denoting gene duplication events. So here and here, these are all in paralogs within one species. And here, it's a gene duplication between species 1, 2, and presumably 3. But here in this part, in this half, species 3 is not present. So we either didn't sample it, the data set is not complete enough, or the second version of the gene was lost. So we select this part of the tree to go forward. So we select these sequences that are marked in bold. And imagine I just shared this with someone, and someone else wants to add his own data to this data set, the species 6. And he gets a nice sequence, only one, it branches here, it all looks correct. So he goes ahead. But if he would add it to this version of the tree, where we have all the orthologs included, he might find out that the species 6 actually branch within this paralog that we don't want to include, right? And that will, because here there should be another one, basically the other paralog of species 6, again, that we don't have in our data, either because our data are incomplete or because the gene has been lost in the genome of the species 6. So here we would see that this sequence is an actual paralog, and we should include it in the data set, but here we don't see it. And it might not make a lot of difference here, but imagine that this data set is being used again and again and again and again. And we get to the 6th generation of the data set, and every time you only choose the already selected orthologs, you snowballing on effect, and eventually you become with a very wrong tree, which is full of paralogs. And that's going to be a problem. So the best way to publish your data set is always publish these data sets along with the ones where you already selected the orthologs. So people that want to reuse the data set, or just want to check your work because maybe they're getting different results, so they want to know if they made a mistake or maybe you made a mistake, they can look here at the original data and see exactly what decisions you made about each gene. And most of these problems we are trying to solve with PhyloFisher. So first of all, PhyloFisher is easy to use for non-bioinformatician experts. It's basically few command lines, commands, and everything happens on its own. It forces manual inspection of trees to search for orthologs with a nice graphical user interface tool to do it. So it makes it very easy. Well, as easy as it could be for the user. It encourages, basically forces people to save their different stages of the data set so they're easy to share for the future analysis and share during the publication. And also it ships with a ready-to-use data sets for eukaryotes. So a lot of people will be just able to take the data set, put in their new transcriptomes, and it will automatically add them to an existing data set, or they can generate their own data set. It can be run in both fashions. And it includes tools that automate the post-assembly analysis. So people can just say, I want to do gene resampling, I want to do fast-evolving site removal, I want to do fast-evolving tax removal, I want to do looking what happens when I remove heterotachyus sites, I want to do what happens when I do amino acid recoding to remove compositional bias, etc. That's all there, ready for them to be run very easily. So it basically does all of this. It's a full package for the complete phylogenomic analysis. And here is a little scheme how it's done. So it has a little tool, I believe, for looking for alternative genetic codes, which is not which is optional. And then it takes per-acute proteomes, build a starting database, and basically search for the genes of interest using Hammer or BLAST or etc. It computes some basic statistics about the data sets. It looks quickly for known deep orthologs and generates single gene trees, which are then passed to the user, who kind of has to open them and approve and basically do or fix the paralog ortholog selection. I'll show you this in a little bigger window in a little bit. One that is done, it will automatically save TSV files with definition of each of the taxonomy tree, whether it's an ortholog or paralog, and it updates the database and select the orthologs, redo alignments and trimming and everything else, and builds a phylogenetic supermatrix that is then returned to the user for the final tree computation. After that, user can come and use any of these post assembly analysis like fast-evolving site removal, gene resampling, amino acid composition, etc., etc. Really quickly, I just want to show you a highlight of this. When you use it, it looks like this. It runs, I think, in like a Chrome-like environment. And basically, this is a tree that you're going to get back. This is just a zoom on a piece of the tree because the tree tends to be fairly big. There is some basic information about what gene you're looking at, how it's long, etc. Are there any suspicious clades? We have an automatic check for when we know, for example, the taxonomy of the organism. It will give warning if it branches somewhere completely wrong. Like, for example, if an animal is branching within, I don't know, plants, that would imply, a chance of contamination, or when there's a very high support for a relationship that we wouldn't expect. So it just gives you warning to look at it. Then all the taxa are basically colored by taxonomy that you predefine in your database. And there are these little squares that you can basically tick by clicking on them. And the green one you click if you want this marked as an ortholog. Yellow one you click to paralog, and red one you click when you want to delete the sequence forever because you're saying it's either contamination or something. So it gets fully removed from the database, while the paralogs that are kept for the subsequent analysis. So when we're adding more and more data to it, we know what decisions we made about these sequences later. Now I realize I probably didn't tell you the most important part, that we're not selecting during the procedure when we're selecting the sequences. Obviously, we're not selecting the best hit. We're selecting X best hits, 5, 10, 15 per taxon to get as good picture of the gene duplication events as possible. So this has been published very recently, about a month ago, in PLOS Biology. It was kind of a complicated publication because we aim this software to be used on sort of a deep relationship, saying, it can be used on any kind of data. Basically, whatever data you have, you can feed it into this software. As long as you have your proteomes and genes of interest, it will do it. However, reviewers didn't believe us and forced us to do this on an entirely different scale. So here, in our original paper, we look at the whole eukaryote, so very, very deep relationships. And then we also looked into evolution of Saccharomyces lineages in collaboration with Antonis Rokas and Xin Chen. And they did this before they helped us with this. And basically, their phylogenomic data set published in Cell, we basically reanalyzed it partially, and we identified several paralogs because we looked at the tree by eye, while at the time they didn't. So they were pretty excited about this as well. And what we would like to do with PhyloFisher is to implement it as a web service. So even the people that don't know how to use anything about command line, which is a lot of people that will now want to do phylogenomic analysis because the sequences are easy to get for many organisms these days. We would like to implement the web service implementation. And here I kind of colored the different steps. So the stage 1 is the initial data set assembly. That's basically done fully automatically. And then there is the stage 2 when the single gene trees are constructed. This can either be done as part of the web service, or if people don't want to wait for it, because obviously we're not going to have available hundreds of CPUs, they can get the single gene tree alignments and run the trees on their own if they have access on their own computation clusters where it is available. Then ortholog selection, that one has to be done by the users. Again, we can implement a user interface online, or people can download the trees, do it on their own computer with our software, and then return it back to the website. And then again, stage 4, when the final data set is done, when the orthologs are selected based on the user wishes, and the final data set is produced, that will be fully automatic. And then the stage 5, I have here in green because they can be used, as needed, and it also can be used on data sets that were not assembled using this web service. So basically, any concatenation can be pushed into this this part of the web service, and it will generate the alignments and data as is needed. Now, the tricky part, the most tricky part about a web service would be, and optimally, and this is very, this is maybe too much, but I think it would be great, would be to have the web service 2 versions of it. One would be a public data set, where all the public data are available. So when someone wants to use our web service with PhyloFisher, he'll upload the data here to the to his private user, and he gets the public data, and his data get up to this public data and analyzed. And once they get published, and they keep being private until they're published on the server, and when they get published, we move them into the public repository. So every time a data set gets published, the public repository gets updated with the newest data. So the users, when they're getting into this, they're getting the most recent versions of their data sets, whether it's our eukaryotic data sets or whether it's a data set that some user assembles on its own using the web service, and then it just kept being stored there and reused and reused and reused. So it could be a sort of like a not only a web service, but also a repository for these data, which would make it would basically enforce, really fully enforce the data sharing, because sometimes it's not really easy to convince people they should share their results. Yeah, so that's I think it for me. I would just like to thank everyone. I especially Alexander, Thijs, all these guys, the first up guys, they basically did most of the footwork. Matthew Brown for making me a big part of this paper since we started working on it together. All the other coders, the paper has a lot of coders. Ruda Vohnot for organizing this, and Irka Peck for helping me and encouraging me to put this in ELIXIR as a web service. For Jiří Macas also being very encouraging, giving me some salary for a postdoc. Obviously IT4I, because they help us a lot while running some of the trees, and ELIXIR. And you guys for listening. I hope it wasn't too chaotic. Thank you very much. I'll take question.
Thank you very much for a nice talk and for a very nice introduction to the field, which was quite easy to follow for non-experts. And we have two questions. One from Marian Novotny. It has been shown that careful construction of alignments can be used to improve AlphaFold models. Could your tool be used to generate sets of orthologs of genomic sequences for AlphaFold modeling? So I understand that it's about like some coevolution estimation, some correlation between positions in alignments. If this
I'm not sure what alpha modeling means, to say the truth. However, I think if you need a good definition of orthologs and paralogs on a tree, yes, it could be the tool. The first part of the tool basically just finds possible homologs. Then it filters them out for their obvious deep paralogs, and then it basically just builds the tree, and it's up to the user to mark their ortholog paralog selection. So if that's what would need to be done, then I think sure, yes, it would. And I hope it's helpful.
Thank you very much. And there is a question from Marek Schwarz. So despite the paper is for eukarya, the package is usable for anything, e.g. bacteria?
Yes, absolutely. Though bacterial phylogenomics, I've done it only once, and it was very, very messy. So theoretically, yes, it should be usable for anything, because the mechanisms are going to be the same. With bacteria, the problem you're going to run into is not so much maybe paralogy, even though the two, but mostly lateral gene transfer, because as far as I understand, the gene exchange between different lineages is very common, and it might be very hard to resolve on the single gene trees and select the data that basically follow the organism path versus the one that followed all these genes was transferred from beta proteobacteria to firmicutes, for example.
The question continues: would I need to provide some extra external data, or would just selecting genomes or having the sequences of interest suffice for the analysis?
Having the genomes or transcriptomes or any proteome basically is sufficient. You want to have proteome. You want to provide protein data. That's the starting part. So predicted genome is good enough. And you also need, you can either use our data set, so the genes we already have, there's about actually 250 genes, or if there's a data set you want to go by, you just provide the reference sequences for each of the genes as a starting part.
So I think both questions were answered, and those who asked are very happy. And thank you very much again for a very nice talk. And I look forward to see all people next week.
Thank you.
So, goodbye.
Bye-bye.